the second part of the lecture, I'm going to concentrate on hemoglobin and myoglobin. So myoglobin consists of a globin, the polypeptide chain, with a heme group. It exists in heart and skeletal muscles. Its function is to work as a reservoir, a storage, and the oxygen, oxygen carrier protein in these tissues. Above there, you can see the structure. This is the three-dimensional structure of myoglobin. What you see in yellow and green is the polypeptide chain. What you see in the other, that stick model is the heme group. Okay. Hemoglobin. So this is myoglobin. Myoglobin has primary, secondary, and tertiary structure. It doesn't have a quaternary structure. But hemoglobin has all 4 levels of structure. So here, this is also the true three-dimensional structure of hemoglobin. It is represented here in different colors. Why? Because it's showing the 4 subunits. So 4 polypeptide chains. Every 4 polypeptide chains made of hemoglobin, after they've been made, every 4 come together to make one mature hemoglobin molecule. Another 4 come together to make another, and so on. Each subunit contains a heme group. I'll talk about the heme group in a minute. So hemoglobin has primary, secondary, and tertiary and quaternary structure. The quaternary structure is the coming of the every 4 polypeptide chain to come together, to work together, to function together. So it's like hemoglobin, it's like myoglobin, but myoglobin, every molecule, single molecule of myoglobin works on its own. It doesn't need to work with other molecules of myoglobin. But hemoglobin, it's like myoglobin, but every 4 together, because the structures are also, and also the functions are very similar. It consists of many alpha helices on a secondary level, and it has a spherical structure in general. Okay, this is heme. Which one is heme? This one in black. This part is the heme group, including the Fe+2 in the middle. أما أمرشا أما heme group here. What is shown in green and here this one in light blue, these are the amino acids of the polypeptide of hemoglobin and, in the case of myoglobin, that bind to the heme group. The heme group must be bound to some of the amino acids. That's how it stays in there. The heme group is not composed of amino acids, so it's not proteinous part. It's a small organic compound composed of pyrrole rings. What you see up there is oxygen gas, O2. The way the oxygen binds is critical, is extremely important. Look at it. One oxygen atom of the oxygen gas binds to the Fe of heme in hemoglobin. The other is sticking at an angle. Why is that? Because the pocket of binding, Shirley binding the binding site of hemoglobin is a narrow pocket. It allows the access of only one oxygen atom. Okay? Right. This is important structurally for many things. For the release of oxygen, once the hemoglobin reaches the tissues, and for other things I'm going to talk about. Right. So when it is bound to oxygen and when it is without oxygen, there is a slight conformational change in hemoglobin structure. So it's going to be more relaxed when it's bound to oxygen, and more tense when it is non-bound. So the deoxy form is the T form, and the oxygenated form, here, this is hemoglobin, is the relaxed form. And the binding site is going to be a little open when non-bound, a little more closed when it is bound to oxygen. Right. Because hemoglobin, in the case of hemoglobin, this is particularly important. So, well, why? Because in myoglobin we have just one subunit. So, and this myoglobin, just one subunit, it has one heme group, so every molecule of myoglobin can only carry one oxygen gas atom, gas molecule, sorry. But every hemoglobin molecule, because it composes of 4 subunits and each subunit has a heme group, every one molecule can bind to 4 oxygen gas molecules. Okay, 4 oxygen gas molecules, not oxygen atom, oxygen gas, and O2. The binding affinity of hemoglobin for O2 is increased by O2 saturation. What does it mean? It means when one subunit of hemoglobin binds the first O2, that encourages the other 3 subunits to bind. It induces them to bind oxygen. The second one binds oxygen, the third one binds, and the fourth one. So this is why what you see in this diagram here, when the first binding starts, it encourages the saturation. Tierbuni hemoglobin, the oxygen, encourages the saturation of hemoglobin for oxygen. This curve will come in the study session as well, and we'll talk about it 3 times here. The curve itself, as I told you before, any curve. When you want to understand it, have a look at the y and the x-axis and see what is it measuring. It measures the partial pressure of oxygen on the x-axis and the percentage of saturation of oxygen on the y-axis. So the more pressure of oxygen, in other words, the more oxygen you have, the higher the saturation. ċeni oxygena kazjatir bi, pressure kazjatir ċka oxygena kazjatira, awana saturation zjatir bi. This is the first thing to understand from this graph. Alright. The other thing is, here the shape of the curves normally, this is in normal cases, for myoglobin and hemoglobin are different. Why is that? Look at myoglobin. This is myoglobin, this one here. Myoglobin goes up sharply. Why? Because it has just one binding site. So this binding site either non-bound or bound to oxygen. So once it bonds, it's already saturated. Okay? Hemoglobin, because it has 4 subunits, the shape of the curve is sigmoidal, a little bit like S. It's sigmoidal. And that means, that actually indicates that there is, if you like, a gradual saturation of hemoglobin to oxygen. One subunit, then another subunit, then other subunits, induced by the first binding. Both graphs reach up there and it's flat. Why? Because when they reach 100%, that's it, they're already saturated. They're already full of oxygen, all the binding sites. So that's why it goes flat. No matter how much more oxygen you have, they're already saturated. What are the factors that affect this graph? So, in other words, what are the factors that affect hemoglobin binding to oxygen, increase or decrease binding? Of course, the PO2, so the pressure of oxygen, in other words, the amount of oxygen available, the pH of the environment affects it a lot, and of course CO2. CO2 level also affects it. Okay? We'll talk about all of this. There is this compound called 2,3-BPG or BPG. It's either called bisphosphoglycerate or diphosphoglycerate. It's the same thing. This is a small compound, a small organic compound. It's non-proteinous. A small compound that binds to hemoglobin. What does it do? It exists naturally in RBCs. It decreases the affinity of hemoglobin to oxygen. How? It binds a deoxyhemoglobin and stabilizes it. In other words, when one of the subunits, so hemoglobin, must bind oxygen at the lungs, must be saturated with oxygen at the lungs. When it goes to tissues, it must release the oxygen. This is the whole idea. So hemoglobin must be able to bind it, be saturated, and also to release it at tissues, because it delivers oxygen to the tissues where it's needed. So once hemoglobin gets to the tissues, the first subunit releases the oxygen, that induces BPG to bind hemoglobin in another part, not in the part of oxygen binding. It binds the globin itself, and by this it encourages it more to release the rest of the oxygen. Okay? Now, any idea what other factors? I've already mentioned it, but can you think of how those other factors, or why is hemoglobin induced to release the first oxygen from the first subunit, for example? Yeah.
Concentration gradients?
Yes, concentration effects. Yes, a lot. Concentration of ions, hemoglobin itself, and between the interstitial liquid and plasma, and between that inside the cell and outside the cell. But what else? Well, CO2 has a role. CO2 is already made in the cell as a result of glucose hydrolysis, glucose breaking down, and other biochemical processes. So CO2 binds the hemoglobin not in the place of oxygen, but it binds to it in other place, and that also encourages the release of oxygen. Also, the fact that oxygen gas molecule, the way it is bound, only one oxygen is bound to the heme group, the other oxygen is sticking out. Okay, that is also structurally, geometrically, also an encouragement for hemoglobin to release oxygen. So once it releases the first oxygen, this compound binds and it encourages us to release the rest of it. Okay. This graph here shows the effect of all the factors, and with them temperature as well, on the release and the binding of hemoglobin to oxygen. Again, we have PO2 pressure measured in this unit, and we have the oxyhemoglobin, so again saturation. So it's the same graph. So generally, this is the sigmoidal shape I've explained. This graph, this curve, can shift to the right or to the left based on the conditions. So as you can see up there, if it shifts to the left, it means that with less pressure of oxygen, less amount of oxygen is going to be saturated. So it's going to be saturated quicker. Okay, so decreased temperature, decreased DPG or BPG. Why? Because BPG encourages hemoglobin to release oxygen, not to bind it and to be saturated with it. Okay? And we also have decreased H+. Decreased amount of protons. So that means a high pH. Okay? pH movements. CO also affects it. I'm going to talk about CO also. When it shifts to the right, reduced affinity, so shifting, if the graph is shifted to the right, what will affect it? What will make it, the graph to shift to the right? Increased temperature, increased DPG, because DPG encourages hemoglobin to release oxygen, release it, and increase H+. In other words, in more acidic environments. Why is that? The effect of pH is through the bicarbonic buffering system, which I'm going to mention. So, for example, the release of O2 from hemoglobin is enhanced. The release is enhanced when the pH is lowered, we said, okay? Or the PCO2, the pressure of CO2 is increased, so the more CO2. This is seen in exercising muscles. This is what happens in exercising muscles, and this is why the release of oxygen is enhanced. Why? Because your body, your muscle tissues and cells require more oxygen. So they require a quicker delivery of oxygen to them because they are using energy. What is oxygen needed for, by the way? Oxygen Anyone? Cellular respiration, which is what? What, sorry? Anyone? Yes? Yes, formation of ATP. By what? Yeah, it's breaking down glucose. Oxygen is Oxygen is required to break down glucose in the process of cellular respiration. Bebe oxygen glucose نشكل ريتوا. Glucose نشكل ريتوا ATP درس نابه. كاركاني بروسكاني كابيستي بر ATP درس نابه. يعني جبه جنابه. Okay. So here the oxygen dissociation curve of hemoglobin is shifted to the right, as you can see here. It just shows this is normally 7.2 and 7.6 lira O patient. Decrease in pH results in decreased oxygen affinity, okay, of hemoglobin, and therefore it shifted to the right, okay. And here at lower pH, so this is lower than 7.4, and that is above 7.4. 7.4 is the pH of the blood. At lower pH, greater PO4 is required. So more PO4, more oxygen is required to exist to achieve the saturation of hemoglobin to oxygen. Right. Here in this, it's the same graph. We're just seeing the effect of BPG, a higher well, there we have BPG 0. So if you take all the BPG out of the cells, RBCs, so no BPG, there will be a higher, quicker saturation. Both remote and BPG, encourage hemoglobin oxygen berda, كأجر binding to saturation بأكسجين زوتر أبيتو دركتر بريئة. Whereas if you have a higher concentration of BPG than normal, it will lead to the opposite thing. And the one in blue is the normal amount of concentration of BPG. Okay? Again, this is how it affects it. Okay, affects oxygen saturation. The pH of the effect of pH, I've already mentioned it. Talk about it again here. It's the same graph. Concentration of percentage of saturation here, pressure of oxygen. pH 7.4 is the normal curve. When it is the pH is less, it's shifted to the right. When the pH is higher, it's shifted to the right, to the left. So binding CO2 must bind hemoglobin because hemoglobin takes it back to the lungs when it is released. But CO2 also has a function. It's not just a waste material. Has a very important function. Anyway, it doesn't bind in the same place as oxygen. It binds to the polypeptide chain directly. إلى قيبة شري أكسجين كونية. Okay. بقولن أو capital O CO2 أنا أسرع وغلط يكلمنا تو وسرع أنا أسرع C capital O capital total. So CO2 is more readily dissolved in deoxygenated blood, facilitating the removal from the body after the oxygen has been released to the tissue. It means what? It means it encourages the hemoglobin to release first stop unit of hemoglobin to release oxygen. Once that happens, it is ready to bind the hemoglobin and encourages it to release the rest of the oxygen. So basically, once hemoglobin starts releasing oxygen, that is when it is more ready to bind CO2. Okay. CO2 makes the bicarbonate buffering system of blood. So CO2 has what makes the buffer of the blood. Okay? This is important. It is the bicarbonate buffering system in blood, and CO2 produced from the tissue diffuse into the interstitial liquid, and then from that into the plasma and into the RBCs. بيزحمل بيدنبن أنا كان مجارة. Alright. CO2 produced in the tissues get to the blood. In the blood, it binds with water to make carbonic acid. Carbonic acid is a weak acid, and weak acids are what make buffers. Why? Because weak acids do not dissociate entirely in water solution. So they don't all dissociate they don't all ionize. Some molecules of a weak acid will remain in this form, and some molecules will lose an H plus and remain in the ionic form. Between those two forms of the acid, of the weak acid, there is a balance of pH, and that is why they can work as buffers. So to control the pH of blood, in this case, the pH of blood, within a certain value. Okay, so here, this is bicarbonate, this is the conjugate base, and this is the actual acid. Once you take one proton away from it, what is remained is called the conjugate base. This is very important because the bicarbonate buffer system is very important not only for keeping the pH of the blood, for all functions of all proteins exist in blood, but also for delivery of oxygen. When the pH falls, okay, as I've said already, when the pH falls, individuals breathe more rapidly. More rapidly and expire more CO2. When the pH rises, they breathe more shallowly and vice versa. Okay? So the rate of breathing contributes to the pH and contributes to the effect of CO2 on hemoglobin binding or releasing oxygen. Carbon monoxide is another story. It's highly toxic, as you know, and if it exists in high concentration in an area, within minutes that person may die. And the reason is carbon monoxide, not carbon dioxide, binds directly in the place of oxygen. Acheta sheni oxygen gazaka bindaka. Not only that, it has a higher affinity, a higher ability to bind hemoglobin, about 200 times more. Can anyone tell me why? Boom. Stable oxygen is stable. Any idea why? What? Okay, it's to do with the structure, but any idea? Can elaborate a little more on that? Yeah? John? No. What is oxygen gas made of? Let's do straw. Two oxygen atoms bound together. Okay. What is the number there? Yeah, can you answer? Well, you are getting closer. The answer is a little simpler than that. Carbon monoxide is a smaller molecule than oxygen, so it can fit in the binding pocket. Why is it a smaller molecule? Carbon monoxide is composed of a carbon and oxygen. What is the atomic number of carbon and oxygen? He carbon chaneka. Okay, he oxygen? Okay. I got do oxygen bela O2. Do shanzaya. Ekami goretra. Oxygen goretra. So carbon monoxide is a smaller molecule. It can fit in that pocket much easier. Okay? And of course, as you know, the problem with carbon monoxide, it doesn't have a smell. And that is why it's extremely dangerous. Sources of carbon monoxide, what happens often here is using heaters. صوبينوتي كبكاري هنري بتايبتي كخلط هي شودي أنا تابعاني بكاري دينا شو تابعاني دايق السنة يعني بشيمي ككمنالي تاوس برشتيارينيا وهمو درقاو بنجرة ومو بس شوازي جيرتراوا doesn't allow a good aeration. طبعا أنا همو خطرة وخرابة أجا حتى مسألة كربون مونوكسيد نبي زور خرابة بي زحمة بي بند بندلوي. Alright? So أوي كرودا صوبة كسيرة كينو تكيت يا كما بيتوا نالي نتوا so it is burning incompletely. Anything which is burning incompletely will produce carbon monoxide. So there isn't enough oxygen, so it's carbon monoxide made. And because it doesn't have a smell, it happens usually. جاو كسا مشكلة كتشي لكاتي خلطنا الشوان خلطوه وكسو بوني ناية اتر أغاي لنابي سيركي زوجة بيت حالتي مردن. بلان جيجلوا من سيري شم بچيده اما والله تمان او سعدني اخوة سايبيريا ناجين يعني نازانم ام على مبور فيك ومن السمايانا شكو جولا كما كان خويا راهيناوا او دوشتي زيادة خواردني خراب جوليكم وراهيناني كي زور باقين يعني جلي كي زور خط رابينا جلي خراب كبرتو جلي زور كبرت دايك سمعت بي وقيل بكنا وبزانم جلي كردي أصل أمي للتائر جلي كردي لبراهيش نميكا وجلي كردي جلي كي زور قرم نيا بلام وادروس كراوانا نسيتا نشوا طبقي هوا دروس بيلا بيني تشان ليالي كدايما جلي كردي أو قرماي أدا وطبعا أوش دوري زورة كخلقك جولي زور بو خواردني تندرس بو بلام راهينا نيجاش زور دوري. The other reason is car exposed. In places مثلا أوجرا جانيكا closed, اگر او ساری که ایش بکابو فتره کی زور ممکنه کنه کجابه تو هیچ نفسی کی هوا نبی اوش درسته به CO درسته به. And also heavy smokers, it's been found that about 20% of their oxygen active sites are blocked by CO. So, this is for heavy smokers. Okay. Types of hemoglobins exist in a normal human being because hemoglobins is slightly different, the number of subunits, the type of subunits a little bit, in fetal hemoglobin, in embryonic hemoglobin, and they differ from adult hemoglobin. So what I've been describing so far is mostly the adult hemoglobin. In addition to that, we have mutations in normal adult hemoglobin, like in the case of thalassemia and in the case of sickle cell anemia. These are, we can also say they are sort types of hemoglobin, could be types, yeah, they are mutation, could be considered also normal because those people can normally live, but they have some conditions, and in some places they have it, quite a lot, like some places of Africa. In the case of sickle cell anemia, a lot of people have thalassemia here. As I know, pochi thalassemia lere lamula kanere nafezora. But it's because of relatives, a lot of relatives' marriages, and because, as you will see in gene inheritance lecture, the way it is inherited, the genes are inherited, will be affected if there is interrelation, marriages. Okay, so sickle cell anemia. So every molecule, every one here, two yellow and two blue, are basically, this is one molecule of adult hemoglobin composed of 4 subunits. Okay, so normally, this is how they exist, normal hemoglobin. So every 4 subunit making one large molecule of hemoglobin will be close to the other hemoglobins, but not cling to it. They will be all surrounded by water molecules. As I said in last week's lecture, any molecule of anything dissolved in water, interacting with water, it means that every single molecule of it is going to be surrounded by water molecules. But a mutation in one nucleotide of one of the genes of hemoglobin causes the change of one amino acid in hemoglobin. That change causes a dramatic big change in hemoglobin. It causes the molecules of hemoglobin to cling together and make long chains, as you can see here. And the long chains will have this sickle shape, and as a result, because all the hemoglobin molecules in the RBC will make these chains, as a result, it will affect the entire final shape of the RBC. So the RBCs will have this sickle shape rather than the normal spherical biconcave shape. And of course, it will affect their movement in narrow tubings, not circulation. It will cause them to block the blood flow and so on. Finally, okay, شتيكي أما شيء كما وتوا أما يزور ببسيطي وريجرن شتيكي تكنيكية وإذا نمشي تكنيكي بنظري كبوتري وشكا يعني الله بكبدان راوا من يعني مثالتم بقى بمالي توا. All I want you to understand the very basic away التكنيكانا based on chi. يعني based لسر chi لسر شو factor protein كاركن. Okay, you really don't need to understand details. I haven't put much, well, I have hardly put any details here. So electrophoresis are one of the types of techniques which are used to analyze and characterize proteins. Proteins are charged, and when they are placed in electric field, they will move towards the cathode or anode based on their PI, isoelectric point, or the entire charge, which is made based on the single charge of every amino acid is composed of, okay? This is one thing. The other thing is a gelatinous material is usually used in these techniques. Mostly polyacrylamide is used because the important types of electrophoresis, SDS page stands for sodium disulfide polyacrylamide gel. You are not required to learn the entire name, but only if you learn the abbreviation, that's enough. SDS page. This is a technique which anybody who's working in protein research using it quite a lot. Could be more than 2-3 times a day. It gives a lot of information about the purity of the protein you are purifying, and also the characteristics of it and the size of it. How does it work? So it separates proteins based on size. Why is that? Size is because the technique actually uses something called SDS, sodium disulfide, which is a detergent. A detergent is anything that we wash things with. So, for example, a soap is a detergent. The liquid mesh that you use for washing dishes is a detergent. This is a type of detergent. What does it do? It denatures the protein. So you see, protein denaturing has, its advantages in studying proteins, in understanding, experimenting them. So what does it do? It coats the proteins, all proteins. It denatures it, so it opens the secondary and tertiary structure. It makes it a linear polypeptide, and it also coats the protein because itself is negatively charged. It makes all the proteins that we analyze negatively charged. Why is that important? So that they are separated only on the basis of size. And then, once the protein is treated with this, it is placed in an electrophoretic field, and then after that it is visualized by a certain dye. A dye is any liquid that we add to proteins to visualize it, to see it. So this is, the first diagram here shows how SDS denatures proteins and coats it with negative charge. The second thing is it shows the gelatinous materials when it is made in between two glasses, special glasses. جلك درستك يكم جاب ليكويدي أو جاءكي تناول بين الجلاسك شتي بودا أني وكو أو يناليها بيوال وكو شانية لسرو بوي سامبل كانتكي. And then you have your protein samples. You put it in any of the wells there. If you have many samples, you have in all the wells there. And then you connect that entire thing. You put it in a tank. You put buffer in it, and you connect it to to an electrical supply. Once it's finished, the result is the gelatinous material is already set, and these lines represent the proteins. You have also a set of standards of known molecular weights of proteins. You run it and you compare it to know the size of your protein. It's also important to know the purity of the protein. You are not required to learn this for the exam, but you are required to know the basis of the technique and also what SDS do, how it coats the entire protein negatively charged, and that's it. Okay. Isoelectric focusing is another type of gel electrophoresis, and this type is the opposite. It actually separates protein based on PI, so based on the charge. Okay? And we also have native gels. This is an actual gel which doesn't treat the protein in any way. So the protein is separated both on charge and size together. Okay? It gives other type of information about the protein, about subunits and so on. And finally, the two-dimensional page, which is basically combining the IEF with the SDS. So, Avrupa, this is the final Bez Ahmed. Ya dečne derova, ya basa. Ama çanam jarki tambulen bedengen. Okay, so you combine the two techniques. So the gel for IEF gel to separate proteins based on the charge is done first. Then that same gel is run in a way to separate the proteins based on the size. Again, you only are required to know basic separation. If you want references, these two, in fact, this one is mostly for